Welcome to the Deep Dive. This is where we take complex research, often pretty challenging stuff, and turn it into knowledge you can really use or at least grapple with.
Indeed.
And today, we're doing something pretty radical, a deep dive into something fundamental, inside your own cells, actually. Microtubules.
Right. Those tiny tubes.
Exactly.
Yeah.
And if your old biology lessons just talked about them being, scaffolding, cellular highways,
It's the usual picture, yeah. Tracks for transport.
well, get ready for a major rethink. A complete intellectual shift, really.
Because the sources we're drawing on today frame this whole discussion, well, very differently. We're using the unified coherence theory of everything, the UCTE.
UCTE.
And this isn't just a minor tweak. It redefines the whole game. It sees cellular life itself as fundamentally about quantum resonance.
So our mission today, for you listening, is to really unpack what the UCTE is claiming here. It's a pretty explosive idea.
It is.
We're talking about microtubules, MTs, not as passive bits of structure, but as active, complex, what the sources call quantum coherence engines.
That's the core term. Engines of coherence.
Dynamic resonance conduits, always working to what? Restore, modulate, extend quantum coherence, all through the cell.
Exactly, and that's the link.
Yeah.
They argue this directly connects basic cell processes to, well, the bigger picture, consciousness itself.
That connection.
Yeah.
That's the huge paradigm shift you mentioned we need to get our heads around.
Right, because within the UCTE, microtubules are really elevated. They become central players in something called the bioelectric code.
The bioelectric
They're
code.
the crucial go-between, mediating this flow of negentropic information as order between the DNA.
Which provides the blueprint, the sort of fractal instructions.
Precisely, the blueprint, and the wider field of cellular intelligence, which, the theory suggests, ultimately builds up towards awareness.
So their main job, then, is keeping the cell coherent, energetically, informationally, fighting off chaos.
That's the essence of it, actively pushing back against that drift towards entropy, maintaining order.
Okay, let's nail this down right at the start, because this is fundamental. We need to properly define this central term. What exactly is a quantum coherence engine in this UCTE context?
Right, so think about a normal engine, maybe in your car. It does work, converts energy, but usually it increases disorder, right? Entropy goes up.
Yeah, heat, exhaust, chaos.
A quantum coherence engine, though, as defined here, is a biological system specifically designed, you could say engineered, to restore coherence.
Restore it after what?
After it's been disturbed, after quantum states have been perturbed, or what the UCTE calls reduced. It actively fights decoherence.
So it's like an anti-decoherence machine.
In a way, yes. And the microtubule is presented as the prime example in biology. It's constantly self-stabilizing, constantly reestablishing these phase-locked resonance states. Snap! Back into order.
Even within the cell, which is, a messy, noisy place.
Especially within that noisy place. That's the point.
That really implies, well, intelligence in the structure itself. It's
Right.
not just a passive track. It's actively repairing, fighting entropy. Built right in.
Exactly right. It suggests life isn't just slowing down entropy. It's structurally creating and maintaining these pockets of high-order negentropy.
Yeah.
And the microtubule is the key piece of machinery for that.
Okay, so traditional biology looks at MTs,
Mm-hmm.
sees support, sees transport pathways, kindness and motors walking along
That's
them.
standard view, yes.
But the UCTE looks at the very shape, the geometry, that cylinder, the lattice pattern, and says, no, this isn't accidental. It's a specific negentropic design.
Optimized.
Optimized for what? For handling resonance, transmitting it.
Precisely. That specific cylindrical shape is crucial. Why? Because a cylinder, especially one made of repeating units with specific electrical properties, like dipoles.
Okay, chart separation.
Yes, it fundamentally changes how waves behave inside it. It's seen as a negentropic design because it's perfectly shaped to sustain and transmit energy waves resonance with minimal loss.
So a hollow cylinder of just the right size.
Yeah.
It becomes a natural resonant cavity.
Exactly, like an organ pipe, but for quantum waves, in a sense. It's physically structured to trap and hold standing waves.
So the energy doesn't just spread out into the cytoplasm and get lost in the noise.
It gets concentrated, contained. That's the key idea. The sources really stress that MTs function as highly specialized waveguides.
Not just for molecules, but for waves.
Yes, both electromagnetic and mechanical resonance, like sound waves, but also subtler energy forms. This concentration is what allows coherent signals, they argue, to travel maybe microns, huge distances inside a cell like a neuron without fading away.
Okay, and you mentioned the structure, the tubulin dimers arranged in this lattice. It supports something called coherence eigenmodes across different frequencies.
Oh, that's a bit dense, I know. Let's unpack eigenmodes.
Please.
In physics, an eigenmode is basically a system's natural, stable way of vibrating. Its preferred frequencies, if you like.
Okay.
The UCTE says the microtubule, because of its unique geometry, has specific, very stable coherence eigenmodes. These are the optimal frequencies it can resonate at without losing energy. Pure tones, almost.
And the structure helps. The way the tubulin parts are arranged.
Yes. The nested structure, the protofilaments slightly offset, forming a helix, it creates complexity. And that complexity seems to act like a frequency multiplier. Meaning the MT can handle multiple distinct resonant streams at the same time, different frequencies, all traveling together without interfering, like multiple channels on one fiber optic cable.
Wow. So it can process a lot of information simultaneously. Complex, integrated signals.
That's the idea. High bandwidth information processing built into the structure.
This completely changes how we see the tubulin dimer itself. It's not just a brick.
Not at all. It becomes a fundamental resonance node in this huge dynamic lattice, an active component.
Because of its dipole properties, the charge separation.
Exactly. That allows it to oscillate, to vibrate energetically. And critically, these oscillations connect to or couple with two things. The water inside the tube.
Which we'll get to, aqua hell.
Yes, the aqua hell. And also the external bioelectric fields, like from the cell membrane.
But hang on. The cell is warm, it's wet, it's chaotic. If you have billions of these little dimers oscillating, shouldn't it just turn into random thermal noise instantly? A jumble?
But that's where the genius of the lattice design comes in, according to the theory. Synchronization.
Okay.
The specific angles, the way the dimers lock together, it forces the oscillations to be phase locked.
Meaning they all vibrate in step, perfectly synchronized.
Perfectly in step, like billions of tiny violin strings all playing the same note, perfectly timed, even in a noisy room. This enforces macroscopic coherence, large-scale order, despite the thermal chaos around it.
And that enforced order, that phase locking across the whole structure, that's the foundation for charge movement.
It provides the essential substrate, yes, for what the UCTE calls tunneling current.
Tunneling currents, okay.
Because the dipoles are so perfectly aligned, they create these continuous pathways, channels. And electrons, or maybe protons, can quantum tunnel along these pathways with virtually no resistance.
Bypassing the usual barriers.
Exactly. The geometry itself is argued to be engineered to allow this coherent signaling over distances that would be impossible for classical electricity in such an environment. It avoids that entropic breakdown.
Okay, so we've got the lattice structure providing this phase-locked coherent framework. Now let's look inside that cylinder. Traditionally, just water, right? Bulk water.
Pretty much ignored, yeah. Or just seen as solvent.
But the UCTE sources redefine this core completely. It's filled with something they call aqua hell.
Yes, and this is a really crucial concept in the UCTE. Aqua shell isn't just water. It's described as a highly structured, almost quantum phase of water.
Quantum water. Like Pollak's fourth phase water, maybe? Exclusion-zone water.
They draw that comparison, yes, to give a sense of the structuring, but UCTE pushes it further into the quantum resonance domain. It's long-range ordered coherent water, not just structured, but actively participating in the quantum dynamics.
How does it stay so structured inside the MT? Water usually wants to move around randomly.
The tubulin lattice itself acts as the template, the inner surface of the cylinder with all those aligned dipole charges.
Right, the phase-locked dimers.
They force the water molecules nearby to align to form this extensive quasi-crystalline network of hydrogen bonds. That ordered network is the aqua shell.
And it's actively maintained.
Yes, through metabolic energy driving those tubulin oscillations. The energy flow keeps the dipole field strong, which constantly reinforces the aqua shell structure, preventing it from just relaxing back into ordinary, disorganized water. And this structured core then acts as what they call the coherence memory field.
A memory field. That sounds key to resilience, like you said earlier. What are the specific properties? How does it help maintain order or nidentropy?
Well, it seems to have three main functions in maintaining that order. First, a restorative function.
Okay.
If the system gets knocked, say, a protein changes shape nearby, or there's some kind of quantum measurement event, which UCTE calls quantum reduction, the aqua shell network is incredibly robust. It snaps back. It reorganizes its hydrogen bonds almost instantly.
like hitting a quantum reset button.
That's a good way to put it. It rapidly reestablishes the baseline coherence state, like a built-in healing mechanism for quantum information, as you said.
Amazing. What's the second function?
It acts as a nigentropic buffer. This is critical in the noisy cell. It's designed to absorb random thermal jolts, incoherent fluctuations, noise, basically.
Wow.
It sort of soaks it up into its ordered structure without letting that noise totally collapse the overall coherence of the microtubule. It shields the important resonant states.
Protecting the signal.
Exactly, maintaining structure even when things get rough. And third, resonant coupling.
Okay.
The aqua shell acts as the mediator. It transfers coherence between the vibrating tubulin lattice on the outside and the larger bioelectric field of the whole cell. It's the stabilized core that connects the lattice dynamics to the bigger picture.
Got it. So the aquagel is this stable, restorative core. Now back to those tunneling currents we mentioned, the ones traveling in the tubulin lattice itself. Electrons or protons flowing coherently. How do they interact with this seemingly static aquagel core?
But the interaction is absolutely vital. It's symbiotic. The currents flow through those dipole-aligned channels in the tubulin protein.
Right.
But their coherence, their ability to stay as a tight, non-dissipating stream, is synchronized and stabilized by the resonant modes of the aquashell core. The aquashell acts like a guiding field, preventing the charges from scattering off course.
So the aquashell stops the current from just fizzling out?
Essentially, yes. It ensures they propagate as coherence-preserving signals. Without that quantum-structured water core reinforcing the coherence, even a tunneling current might dissipate over the distances involved.
Okay, and this leads to something huge mentioned in the sources of the work by people like Bandyopadhyay, suggesting that the tubulin dimers themselves can act as switches, Bistable quantum switches.
Yes, this is absolutely central to the idea of MTs processing information within this bioelectric code. Bistable means the dimer can exist stably in one of two distinct electrical states, like a 0 or a 1.
A biological qubit, almost.
Potentially, yes. And a coherent tunneling current can flip that state, like writing information, without destroying the coherence.
So the microtubule isn't just a wire, it's like a massive interconnected memory chip, a quantum register.
That's the analogy they draw. A vast array of memory sites built into the structure itself. These bistable quantum switches give it memory-like properties, perfectly suited, they argue, for storing and processing the frequency-coded instructions of the bioelectric code.
And the tunneling currents are carrying the instructions.
They're the coherence vectors, carrying the specific signals.
While the aquashell provides the stable environment for it all to happen.
Exactly. The coherence field that sustains the whole operation.
Okay, let's put this together. The engine in action. Can you walk us through the operational cycle? The collapse buffer recovery cycle. How does the MT dynamically fight entropy using both the lattice and the aquashell?
Right. So picture the cycle happening constantly, incredibly fast. Step 1 is collapse. Some kind of perturbation hits the system.
Like a thermal spike, a chemical reaction nearby, a quantum reduction event.
Any of those. Something disrupts the perfect phase alignment of the tubulin lattice and the ordered aquashell. Just for an instant, the system flickers into a less coherent, more disordered state.
Which normally you'd expect to lead to decay, entropy wins.
Right. But then, immediately, step 2, buffering. The aquashell, with its incredibly robust hydrogen bond network, absorbs that sudden burst of incoherence. It takes the hit.
Contains the damage.
Contains it, stabilizes it internally within its structure. It acts as a shock absorber, preventing a total irreversible collapse into decoherence. It dampens the fluctuation.
Okay, buffered. Then what?
Then, step 3, recovery. Because the underlying geometric structure of the tubulin lattice is physically strong, held by molecular bonds, the dipoles naturally want to snap back into their phase-locked alignment.
They reset themselves.
They reset. And that rapid restoration of the electrical template immediately forces the aquashell, which was holding that buffered energy, to reorganize, too. It snaps back into its high coherence state, reestablishing the original resonance patterns.
Wow. Collapse buffer recovery. Happening constantly.
That's the engine principle. It's continuously performing this cycle, actively restoring symmetry and order whenever asymmetry or chaos tries to creep in.
So the microtubule really is this operator of gradients then, managing coherence and incoherence.
It is. The UCTE actually describes it as a meta-operator. It embodies two fundamental operations at once. The aquashell performs symmetry restoration. It's the coherence field operator, always pushing back towards order. The tunneling currents, on the other hand, represent asymmetry resonance. They are the discrete flows, the specific signals, maybe the reduction events that create specific states.
The field and the particle almost.
In a conceptual sense, yes. And this interplay, this synergy between the continuous field restoration, aqua shell, and the discrete resonance flows, currents, is what sustains the whole process of biological logistics. It manages that crucial quantum interface.
Okay, so we have this amazing engine in the core, maintaining coherence locally. But cells are huge, especially neurons. How does this coherence, this negentropic order, get transported reliably over long distances without just fading out?
That's where another fascinating concept comes in, the microtubule soliton.
Solitons. I've heard of these in other contexts, like fiber optics.
Yeah.
Stable waves.
Exactly. A soliton is a truly remarkable thing in physics. It's a localized wave packet that holds its shape and energy as it travels. It resists spreading out, resists dissipation.
Like a little information bullet.
A coherence packet, as the UCTE frames it, a discrete, self-sustaining carrier of negentropy. And they propose these are essential for sending resonant signals faithfully across those large cellular distances.
That's incredible. A stable, energy-preserving wave moving through a biological structure.
Hmm?
What is it about the microtubule that allows these solitons to even form and stay stable?
Well, soliton formation needs a specific balance. It needs dispersion, the natural tendency of waves to spread out, but it also needs something called nonlinearity to counteract that.
Nonlinearity.
Meaning the medium responds differently depending on the wave's intensity. The microtubule lattice provides this perfectly. The tubulin dimers with their strong dipole oscillations and the complex fields around them, that's a highly nonlinear environment.
Okay.
This nonlinearity fights the dispersion. It keeps the wave energy focus, forcing it into that stable, localized soliton shape. The phase locking helps create the continuous nonlinear medium needed.
So the lattice structure itself creates the conditions for solitons. Now, are these solitons just traveling on the lattice? Or does the aqua shell inside play a role too?
Oh, it plays a fundamental role. They interact deeply. The aqua shell isn't just passive. It actively enhances the soliton.
How?
As the soliton wave travels along the lattice, the structured water inside aligns itself even more, forming these quasi-crystalline corridors right in the path of the soliton. It acts like a quantum bridge.
Guiding the soliton.
Guiding it and more. If the soliton starts to lose a tiny bit of energy or its shape starts to waver, the aqua shell structure can actually buffer that potential loss and feed coherence energy back into the wave.
Whoa. It actively sustains the soliton.
That's the proposal. It makes the soliton, this coupled system, part lattice dynamics, part quantum water dynamics, a quantum hydrodynamic wave.
Which would make them incredibly stable and robust.
Exactly. And that's why they're proposed as the primary agents of biologistics.
Biologistics, meaning?
Meaning the organized, high-fidelity transport of this useful, ordered, energy negentropic coherence. Moving beyond just random chemical diffusion, this is directed, purposeful transport of order.
Okay, so tell us more about what these soliton couriers are actually doing. What kind of negentropic information are they carrying?
They seem to perform three critical functions on a large scale. First, simply energy delivery.
Delivering energy, but coherently.
Yes, distributing usable coherence energy that isn't just raw heat, but is structured, information-rich to different parts of the cell without losing it to entropy along the way. Powering processes with order itself.
That's a huge conceptual shift. Energy itself carrying order.
It is. Second function, information encoding. The specific shape and frequency of the soliton wave can carry coded instructions. Messages.
The bioelectric code again?
Right. And because the soliton doesn't dissipate, those instructions arrive intact, perfectly preserved, even over long distances. High-fidelity messaging. Systemic integration. This is crucial. Solitons are seen as the carriers that link the deepest levels, like the resonance patterns coming off the DNA.
Genomic coherence.
With the cell's boundaries, the membrane biofields, the overall system coherence, they tie the whole cell together into one unified quantum entity.
This really sounds like a direct challenge to the usual understanding of entropy in biology. It's not just slowing decay, it's actively maintaining, even enhancing order as it moves.
That's precisely the concept of negentropic maintenance in the UCTE. Solitons don't just minimize entropy, they preserve and amplify order. And they're selective.
How so?
Only coherent resonance patterns can lock into that stable soliton form. Random noise, incoherent fluctuations. They can't form a stable soliton, they just get dissipated by the microtubule structure, filter out,
So the soliton pathway actively rejects noise.
which positions them as living vectors of negentropy. They embody the persistence that life needs to constantly fight against that universal slide towards thermodynamic equilibrium, towards disorder.
Okay, we've built up this picture of the mechanics, the structure, the aqua shell, the currents, the solitons. Now let's make the jump to function. If MTs are built to handle resonance and coherence so exquisitely, what's their primary role?
It's about regulation, managing that flow of coherence. The UCTE positions them as the crucial resonance middleware for what it calls cellular intelligence.
Middleware, like in computing,
Yeah.
connecting different systems.
Exactly. They are the central execution and distribution network for the cell's intelligence. If you think of DNA as the hard drive with the basic instructions.
The instructional resonator providing the blueprint.
Right, the blueprint, the potential eigenmodes. Then the microtubules are the dynamic processors and the communication bus. They translate those abstract instructions into actual physical moving patterns of resonance. The solitons, the currents coordinating activity across the whole cell in space and time.
So they're not just passively reacting to chemicals floating by. They're actively shaping the cell's internal environment, its electromagnetic fields.
Very actively. They function as complex field modulators in three main ways.
Okay,
First, they act as field amplifiers. They can take very subtle resonance signals, perhaps originating from a specific gene on the DNA, and couple them up, amplify them, projecting them onto the larger cellular biofield, giving local instructions global significance.
Making small signals loud enough to matter cell-wide.
Precisely. Second, they act as resonance filters. We touched on this with solitons. They ensure that only coherent, meaningful signals of the standing waves, the nigentropic solitons, get propagated widely. They block out the incoherent noise that would just scramble the instructions. Quality control.
Makes sense. And the third function, coordination, seems vital if we're talking intelligence.
Absolutely. The third role is phase coordinators. This is key. They synchronize the oscillations, the resonance patterns across different parts of the cell, connecting organelles, linking up with other parts of the cytoskeleton, coordinating with the cell membrane.
Creating a unified resonant field.
Exactly. It's this globally synchronized coherence, orchestrated by the microtubule network, that allows for unified, intelligent cellular action. Without it, the cell would just be a bag of disconnected chemical reactions.
And this coordinated system is what the sources refer to as the triadic coherence system of the cell, leading to cellular coherence intelligence, CCI.
Yes, CCI isn't located in one place. It emerges from the synergy, the coherent interplay of three key elements, each doing its part in the resonance dance.
Let's break down that triad. Element 1.
Element 1 is foundational, DNA, the instructional resonator. It holds the library, the spectral blueprints, defining the cell's ideal resonance patterns, its coherence eigenmodes, the potential states.
Okay. Element 2 is our main focus today.
Microtubules, the execution engines. They take those instructions, those eigenmodes from the DNA, and make them real. They translate the plan into actual resonance flows, solitons carrying energy and information, tunneling currents, switching states. They perform the symmetry restoration and distribution. They execute the plan.
Making the abstract physical. And the third element has to connect outwards.
Yeah.
Right?
Yes. Element 3, the membranes. Primarily the cell membrane, but maybe organelle membranes too. They act as the projection fields.
Projecting what?
Projecting the cell's internal integrated coherent state outwards, translating it into bioelectric signals and fields that interface with the outside world, the extracellular matrix, other cells, the organism's overall field.
So a constant loop. Instruction from DNA.
Execution and distribution by microtubules.
Projection and interface by membranes. And that cycle is cellular intelligence.
That recursive coherent triad is CCI, according to UCTE.
This is way beyond the old view. It's a massive ontological shift, isn't it? We're not talking about a biochemical machine, a complex clockwork reacting to chemicals.
Not at all. The sources really insist on this. The cell becomes a coherence participant, an intelligent entity whose actions, whose agency stem directly from these resonance mechanics. Chemistry becomes secondary in a way, guided and constrained by the underlying coherence field.
Wow. And that core axis, the DNA-microtubule link, it's not just one-way traffic.
No, critically, it's bidirectional, a recursive resonance dialogue. The coherent state of the microtubule network feeds back, influencing the DNA environment, potentially tuning gene expression, affecting epigenetic states in real time.
So the cell's intelligence isn't fixed or just stored chemically. It's dynamic, constantly tuned by resonance, responding instantly.
That's the implication. Response is far faster and more integrated than chemical signaling alone could ever explain.
Okay, this is already mind-bending at the cellular level. Cellular coherence intelligence, CCI, driven by this DNA microtubule resonance. But the really big provocative claim is how this scales up. How do we get from one cell's intelligence to, well, us, to cognition, subjective experience?
Right. This is where the UCTE framework reaches its peak, you could say. The focus shifts to the brain, specifically to the neurons.
With their massive arrays of microtubules.
Immense arrays, particularly in the long axons and complex dendrites. The theory proposes that these neuronal structures take CCI and scale it up dramatically into networked coherence intelligence, or NCI.
NCI?
And this NCI is proposed as the fundamental substrate of cognition. Neurons are perfectly built for this, with their long shapes creating extended corridors for coherence to propagate and connect over macroscopic distances in the brain.
But the brain is the ultimate noisy environment, isn't it? Neurons firing like mad heat, constant biochemical churn. How can delicate quantum coherence possibly survive in there, let alone be the basis for thought?
This is where the aqua gel concept becomes even more critical, scaled up to the neural level, that negentropic buffering function we talked about.
Yeah, absorbing noise, restoring order.
In neuronal microtubules, it's proposed to be even more robust. The quantum water structure is supposedly maintained even under intense metabolic load and rapid electrical firing. It constantly buffers the noise, preserves the resonance fidelity during action potentials, stabilizes coherence during the messy process of synaptic transmission.
So aquashell is the key enabler for quantum effects in the hot, wet brain.
It's presented as the essential stabilizing medium. Without it, any quantum coherence would likely decohere almost instantly.
And the sources push this even further. They suggest this stability allows for quantum entanglement between microtubules in different neurons.
Yes, that's a major claim. The idea is that the stable aquashell environment allows for non-local correlations entanglement to be established and maintained between microtubules in neurons that might be physically quite far apart in the brain.
Creating non-local coherence bridges.
Exactly, linking different brain regions instantaneously, non-locally. This is offered as a possible physical basis for the feeling of unity in consciousness, the binding of different sensory inputs into a single experience. Aquashell becomes the hidden substrate supporting this global synchronized network activity.
Integration that happens faster than nerve signals could travel classically.
That's the argument, something beyond just electrical pulses.
So maybe the action potential we measure is just the output, the downstream effect. But the real processing, the cognition itself, is happening inside in these coherence dynamics, powered by those solitons.
That's precisely the proposal. Those non-dissipative soliton waves traveling along the neuronal MTs are suggested to be the actual carriers of higher cognitive functions. Far faster, or far more complex information processing than action potentials alone could merit.
Okay, let's get specific. This is staggering. What cognitive processes are these solitons supposedly carrying? What's the payload?
Well, the theory links them to three fundamental aspects of cognition. First, memory encoding. Solitons, being stable resonance packets, are proposed as coherence engrams, physical patterns of resonance holding the memory. Because they don't dissipate, the memory trace could be incredibly stable and long-lasting, stored energetically, not just structurally.
Okay, memory. What else?
Second, perception binding. How do we experience sights, sounds, touch all together as one unified moment? The theory suggests solitons traveling through interconnected neuronal networks synchronize the activity across different sensory processing areas, binding it all together into a single conscious frame.
Synchronizing distributed activity through coherent waves. Makes intuitive sense. And the third one, this is where it gets really deep, qualia projection.
Ah, qualia, the raw feel of experience. The redness of red, the sharpness of pain, the hard problem.
Exactly. How does physics create subjective feeling?
The UCTE offers a mechanism, speculative of course, but a mechanism. It suggests that when these coherent solitons interact within the microtubule arrays and complex patterns, they create, well, they call them resonant holograms.
Holograms, like light.
Not light holograms, more like complex self-organizing fields of resonance. The frequency codes carried by the solitons when they interact and interfere within the MT lattice project this unified, incredibly complex internal resonance field. And that field is proposed to be what we experience as subjective qualia. The pattern is the feeling.
The resonance pattern is the redness of red.
That's the radical idea. Qualia as emergent properties of highly ordered microtubule-based resonance fields.
This leads us to the ultimate role UCTE assigns the microtubule, the meta-operator of consciousness. How does that internal cycle we discussed, collapse, buffer, recovery, relate to awareness itself?
Okay, so consciousness in this framework isn't a thing or a place. It is the process. It's the emergent resonance of these coherence cascades.
The dynamic itself.
Yes, and the microtubule embodies the engine of that dynamic because it's constantly performing two fundamental complementary operations, the two universal operators needed for experience to arise.
Which are?
First, the symmetry coherence operator. That's the aqua buffering, the phase-locking restoring order, maintaining the background coherence field. Think of this as the underlying potential, the unified awareness, consciousness as field, pure potential.
Okay, the ground state. And the second?
The asymmetry resonance operator. This is the action part. The tunneling currents creating specific states, the solitons carrying discrete information packets. This is the process of reduction, collapsing the potential field into specific, definite percepts or thoughts. This is the observer function, the moment of actual experience.
So consciousness is the constant interplay, the dance, between this underlying unified field being momentarily collapsed or reduced into specific experiences and then immediately being restored.
You've got it. That's the core idea. Neuronal microtubules are relentlessly restoring the background coherence, symmetry operator via aqua, etc., while simultaneously using that stable platform to generate specific, discrete states of awareness, asymmetry operator via solitons currents. Consciousness is that fundamental coherence dance between potential and actuality, enacted moment by moment within these engines.
And this connects everything, from the smallest scale upwards.
Yeah.
The nested cascades concept.
Yes, it provides a hierarchy, a bridge across scales. It starts at the cellular level, DNA-MT interactions creating basic CCI.
Right.
That scales up seamlessly via neuronal arrays to the neuronal level, generating networked coherence intelligence, NCI, enabling entanglement.
Okay.
This then manifests at the cognitive level as the unified subjective awareness we experience, global brain coherence.
And the UCTE pushes it even further.
It does. It suggests this biological coherence system ultimately participates at the cosmic level, that our consciousness resonates with or taps into underlying universal coherence fields, what the theory calls the zeta naught potential, the base field of reality.
So the microtubule, it's literally the bridge, connecting molecular resonance all the way up to potentially cosmic awareness.
That's the profound and admittedly speculative implication of the entire framework. An ontological bridge built into our biology. #outro.
Wow, okay. This deep dive has definitely forced us way outside the standard textbook view of the cell. We've gone from seeing the cytoskeleton as just, boring passive scaffolding
Humble support structures.
to understanding it, or at least considering it, as this active, intelligent, resonant framework, something that could be driving life and even consciousness itself. It's a lot to take in.
It absolutely is. If we synthesize the UCTE view, the microtubule really plays three core intertwined roles. It's a coherence regulator, using aqua, using its structure to constantly fight noise, maintain order, amplify the right signals. It's also a soliton corridor, a quantum pipeline transporting these high-fidelity packets of energy and information and entropy across the cell using those amazing quantum hydrodynamic waves.
Okay. Regulator, corridor?
And ultimately, potentially, a consciousness bridge, cascading that coherence up from the cellular level through neural networks to give rise to the unified awareness of the whole organism.
And the architecture itself, the cylinder, the lattice, the aquashell core, it's not just clever mechanics. He said it's the physical embodiment of entropy.
Yeah.
An ontological framework.
That's how the sources frame it. The structure is the principle of order made manifest. And because of that structure, the UCTE proposes something called the conservation of coherence principle in biology.
Conservation of coherence. Meaning
That the system is fundamentally built to win against disorder, locally at least. The sources argue that even if some major perturbation hits and causes a temporary collapse into decoherence, the inherent design of the microtubule, the buffering of the aqua, the self-stabilizing nature of the solitons, it guarantees the system will snap back. It will restore symmetry, reestablish the ordered state.
order is guaranteed to reemerge from chaos because of the structure itself.
That's the principle. Resilience built into the quantum foundations of life.
That is definitely a provocative thought for you, the listener, to take away and wrestle with. Consider what that principle really implies. If the very structure of life, down at this fundamental microtubule level, is engineered to guarantee the restoration of order against the constant tide of universal entropy, What does that say about life's persistence? Is consciousness then not just some random evolutionary fluke, but maybe a structurally, quantum mechanically inevitable outcome of a universe built with such principles? Something to ponder.